Mike is a associate professor of medicine and internal medicine and medical oncology. He shares patients, or he cares for patients with genitourinary cancers as part of the Prostate Neurologic Cancer Program. Prior to joining Yale in 2009, Dr. Hurwitz was an instructor of medicine at Harvard and an attending physician in medicine at the Massachusetts General Hospital. Mike's a graduate of Harvard College. He received his doctorate degree in cell biology from Rockefeller University, as medical degree from Cornell University. He completed a fellowship in oncology at Dana-Farber and a postdoctoral fellowship in biology at the Massachusetts General MIT institution. Mike's going to be talking about the Yale cell therapy program for solid tumors. Mike, take it away.
Thanks, Dan. Yeah, thanks everyone for inviting us from the cell therapy to talk. I'm going to talk obviously about the solid tumor side, and then the other half is going to be Isufi talking about liquid. So we're the newest DART, and we really started right before COVID. So we don't have a whole lot of trials open. So I think that this talk is going to be sort of short on data, but I hope it's going to be long on potential. Okay, let me see if I can move my thing forward. Here are my disclosures. So the main therapies I'm going to talk about today are CAR T cells and TIL, or tumor infiltrating lymphocytes. And I think that everybody is somewhat familiar with these terms. I know that a lot of people know a lot about these so far, but they're really quite different therapies. And I do want to talk a little bit about the basic biology. So for those who are immunologists, bear with us, maybe read the newspaper for a minute or two while I give you my very simple oncologist view of immunology. So adaptive immunity is where T cells primarily recognize things that are foreign and use and attack them. Now, one of the reasons we don't attack ourselves is that we are always taking little chunks of our proteins, expressing them on the surface in something called the major histocompatibility complex. And the T cell receptors, basically, when we're in utero and a little bit after that, all the T cell receptors that we have, the T cells that recognize, that recognize the antigens well, get deleted, or at least they get turned off. Okay, so generally we don't respond to our own antigens. But if you get a foreign antigen, like a bacteria, what happens is, let's say if they go into a cell, the cell chops up the proteins get put on MHC, and the T cell receptor is going to recognize those. There's going to be a strong interaction. But that isn't enough actually to cause killing. It's only when you get something called costimulation, okay? And that's via another pathway, another set of receptors, and then you actually get killing, all right? So how can we use that information to kill cancer cells? So let me say a little bit more and go a little bit more in depth into T cell receptor signaling first. So this is a schematic of the T cell receptor. The alpha beta chains are the ones that actually recognize the antigens in MHC. And then there are signaling molecules, the zeta chain and the associated CD3. So T cell receptors only recognize proteins. They only work if the antigen is presented by MHC, and they require co-stimulation. And as I said, the signaling are through these two things. Antibodies, another way that we recognize things that are foreign, work quite differently. They can recognize any type of antigen. It doesn't have to be protein. They don't use MHC, and antibodies are much, much stronger, their interactions with their antigens, than T cell receptors are with their antigen MHC interactions, up to 1,000 to 10,000 fold stronger, in fact. So someone had the bright idea of taking the back end of a T cell receptor and connecting it to the front end of an antibody. And we call those chimeric antigen receptors. And so this is the first generation CAR. And this was actually in the 1990s. It was a while ago. So this is the antibody, okay, on the outside of the cell. And this is part of the T cell receptor on the inside of the cell. It worked a little bit, but not terribly well. A huge breakthrough, though, came in the second generation. And here what was done is they added a domain to the protein of CD28. And what's that? Whoops. That, of course, is the co-stimulatory signal. And so when you put the co-stimulator right in it, these are much, much more powerful. These are really what we mostly use today. There are even stronger ones, the third generations, that use two co-stimulatory signals. And there's the fourth generation, which is a combination of CARs plus other genes that are put in to make the cells work better. So when you actually, the mechanics of this in patients, are complicated, just like all cell therapies are, whether it's transplant or something like this. In this case, you need to isolate the T cells. They have to get activated, and then they're transduced with the chimeric antigen receptor, and then expanded, and then reinfused. In the meantime, patients get lymphodepleted. And the reason for that is that there are probably twofold reasons. For some reasons, you're actually treating the cancer to some degree by lymphodepleting. But that isn't the case for all cancers. For some, you're doing it to have a niche for the T cells to actually live in and grow in. As you might imagine, this does not take a day. This takes several weeks. So one of the things about this kind of treatment is patients have to be well enough to survive those weeks and to be able to tolerate the therapy. I'm not going to go into this in detail, but there are a lot of toxicities associated with these treatments. The three famous ones are cytokine release syndrome, which has to do with a lot of T cells at the same time seeing antigen and then causing lots of cytokines to go into the circulation. There's also neurotoxicity called CRESS or CANS, and probably the most severe of these, HLH. All right, so what are we doing at Yale? We have one CAR T study open right now for solid tumors. This is a kidney cancer trial done by the company CRISPR Therapeutics. It's anti-CD70, which is highly expressed on clear cell kidney cancers. And then there's some expression on a few lymphoid type of cells. Now, it's a very long name for the trial. And the reason is that it's actually a little more complicated than even what I described before, because these are allogeneic engineered T cells. And what does that mean? So these are T cells that actually don't come from the patient. They come from sort of healthy donors in whom they're having the CAR put into their own T cells. And this company uses something called CRISPR-Cas9. I think a lot of us are familiar with that, to knock out certain other genes in these T cells to make them work in us. So what do I mean by that? Well, if you take someone else's T cells and put them into you, they will attack you and you will attack it. It won't be an effective therapy. They will get destroyed pretty quickly by the endogenous immune system. And they're going to have off-target effects too, but via their T cell receptors potentially. So what they've done, this CRISPR Therapeutics, is in addition to putting in the CAR to these T cells, they've also put in, using CRISPR, they've removed the T cell receptor, okay? They've also removed something called beta-2 microglobulin, which is part of MHC class 1. And the result is that our immune system doesn't recognize that it very well, except by something called natural killer cells. It doesn't do that much. And it doesn't really recognize us except via the anti-CD70, all right? And so there are a bunch of advantages here of using T cells from someone else and not from the patient. One is speed. These cells are waiting. The patients don't have to wait. Secondly, these cells are for someone with an intact immune system. And a lot of patients with extensive cancers may not have intact immune systems, and their T cells may be somewhat dysfunctional. And obviously, it leads to the potential for more of a drug, something that can be done with high levels of production out there for everybody. We've enrolled one patient. We're going to be enrolling another patient in a few months. We're at the dose escalation phase, and we'll see how this trial goes. What else is going on at Yale, though? Oh, sorry, before I go there, let me just say that, so what some of the challenges are for CAR T cells, specifically in solid tumors. Well, in general, there can be an issue with persistence of the CAR T cells. They may not last. But these are big issues for solid tumors. So one, there is almost no antigen a solid tumor cannot lose. And when you give something like CAR T therapy against a particular antigen, it's very likely that the tumor will just mutate or lower expression of that antigen and become resistant to it. In addition to that, the microenvironment of the tumor is very toxic to a lot of immune cells, including T cells. It's hard to infiltrate into a lot of tumors. There's a lot of necrosis. Many of the cells have low blood supply, et cetera. And finally, the toxicities that I sort of mentioned before. So one of the things that's being done here is being done by the lab of C.D. Chen. His is the only lab I'm going to talk about, but it's worth pointing out there are many labs here working on CAR T type therapies. The C.D. Chen's lab has developed a modular high throughput way of developing CAR Ts. And it's the system that again, is very complex. There's no time for me to describe it, and B, I wouldn't be able to very well anyway. But this is a slide from CD. But Again, this is his own system that he's designed using adeno-associated virus to make CAR Ts. It enables a rapid building of new modules because it's modular, we can put in many different CARs into a lot of cells and look at them in parallel. And it also allows for knockout of other genes in the cell to try to improve the cell's capabilities. And therefore, what we're looking for is superior cancer killing based on a lot of the platforms that are used now. And the short-term goal, of course, is to generate better CAR Ts against kidney cancer. He's actually looking at kidney cancer, which is great because that's a lot of what I'm interested in. And we're also working with Dr. Kluger on this, Harriet Kluger. But also he can engineer in safety control elements so that if the T cells are causing some of these severe toxicities, they can be turned off. You know, the long-term goal, of course, is just to optimize better CAR Ts across solid tumors and maybe liquid tumors too. And the first step of that, hopefully, will be once his lab develops what he thinks is a great CAR, is for us to actually put it into trials. All right, moving on to TIL. Let's go back very quickly again into immunity. So remember, something foreign is a strong interaction by the T cell receptor and the MHC complex. You get co-stimulation and you get killing. All right, but how do you get a T cell to actually kill something that's not foreign, right? As mentioned before, we don't interact very well with our own antigens. Now, cancer has sort of solved that a little bit for us in that cancer proteins are often mutated because cancer causes mutations, and therefore the peptides actually can look foreign, and so you can actually get T cells to kill. As we all know, though, it doesn't really work very well on its own. We need to give things like immune checkpoint inhibitors because of the toxic microenvironment. So a thought that was began developing back in the 1980s was, well, maybe if we take the two T cells out of that environment, grow them up, enhance their function with cytokines, maybe we can cause cell killing if we reinfuse those T cells. And that's what TIL therapy is. So much like I described with CAR T cells, you resect the tumors from patients, T cells are isolated from those tumors, they're activated and expanded in vitro, generally using interleukin-2, but there are other interleukins we use, and then they're reinfused into the patient. In the meantime, patients have been lymphodepleted, which is extremely important for this therapy, because we not only have to have a niche for the cells to go into, we have to get rid of T regulatory cells, and the immune system acts as a cytokine sink, sucking up all the good cytokines that we want to go to these T cells. Because when we infuse these T cells, we give patients interleukin-2. Now, before I move on with TIL, I think a lot of the time when I tell people that we're interested in cell therapies, they say, oh, it's CAR T cell therapy. But there's a huge differences really between CAR T cell therapies and TILs. And I think of them as really entirely different. As some examples, you know, CAR T cells are MHC totally independent, right? They're using an antibody, whereas TIL therapy is totally dependent. CAR T cells they can look at sugars or other non-protein antigens. TILs do not. CARs are pretty ineffective at looking at intracellular proteins. They're working on that, so maybe we'll get there one day, but right now they can't really recognize a lot of the proteins. And a key thing is that, you know, TIL can look at any antigens that they see. So for example, when we take tumors out of patients and isolate the lymphocytes from those, that's going to be a diverse type of T cells, probably recognizing multiple different antigens. And as I said before, a big disadvantage of CAR T cells is that you can lose the one antigen they recognize and they're useless. And that may not be as big of an issue with TIL therapies. And lastly, their toxicities are quite different. All right, so what are we doing at Yale? We have a trial right now for looking at triple negative breast cancer. This is an IIT that I'm doing with Iovance Therapeutics. This is the first dedicated breast cancer TIL trial in the world. We've enrolled 2 patients so far. And one of the reasons we're interested in breast is that there's a lab here, Tristan Park, who's a surgical oncologist here and expert on breast cancer and on breast cancer cell therapies, who is actually looking at the samples we get, analyzing for the immune infiltrates, and working with us on the trial. Just to say a little bit more about what it actually entails, there's a lot of, for any sort of cell therapy, there's a lot of work that goes into it because these are complicated therapies that require good timing. So, you know, once a patient signs consent, They have to get their surgeries. Only then do you initiate, of course, the TIL culture. Then it's going to be going for several weeks. And once you know the TIL is growing appropriately, only then are you going to lymphodeplete the patient and then infuse the TIL into the patient. And then, of course, as I said, these people require IL-2 afterwards. And they're going to be in the hospital for a lot of this because they're going to be lymphodepleted. And then once they recover, they go home. We follow them. So how might we improve some of these things? Well, I think infusion and resection and isolation, that's not where the money is. But clearly, we can maybe improve activating and expanding these cells and make them better killers. And the people who are the best at growing up and activating these cells, of course, at Yale, are the people at the Advanced Cell Therapy Lab run by Alexei Bursnev and Diane Krause. And they have a huge amount of expertise over many years looking at TIL-type therapies. They've grown up a lot of different cell products for use in patients. And I actually hold INDs for growing melanoma TIL, but of course they actually did it. And we're working together right now to grow up lung cancer TIL for ideally to eventually put into patients. Just quickly to point out, they are very good at growing up cells. This is one experiment in which they actually separated out the PD-1 positive from negative cells and show a lot of expansion in both of them. And this just kind of shows one experiment of theirs that the cells they get are actually quite good. So here TIL they've isolated out, and these are assays for interferon gamma production, which is an assay of it's a surrogate for cell killing. And when you take this TIL and you put them alone, they don't make a lot of interferon gamma. As soon as you put them with autologous tumor, where they recognize antigens in the setting of MHC, they make tons of interferon gamma. And then if you give them someone else's tumor that has MHC they don't recognize, they don't kill. So they're very good at making cells that kill and kill specifically, which is exactly what we need. So what can we do to actually improve things to make these, what are we interested in doing here at Yale to improve these therapies? Well, the cell therapy, the ACT lab is doing experiments to look at adjusting the growth medium that they do it in, different cytokine combinations, different levels of cytokines, and those experiments are ongoing. But it's actually striking how little we know about what happens between when we take the cells out of a person and we expand them. We don't really know which cells get expanded. We don't know whether the T cell maturation states, whether they're more naive or more effector cells, dictate which cells get expanded. We don't know how this concept of T cell exhaustion relates to expansion. And we have very little idea about how homogeneous or heterogeneous the T cell infiltrates are between tumors or between tumor types. So, and can we actually do experiments to figure some of this stuff out? And the approach that we're going to take here, and we've actually begun taking, is to do single cell RNA sequencing and paired with TCR sequencing so that we can follow specific T cell clones through growth and figure out which maturation phenotypes are the ones that grow the best, and whether exhaustion has an effect. And then we're going to do this across a number of subjects. So I should say that that's already being done a little bit. One, it's being done beginning in Sam Katz's lab by Sam Kerr, one of his graduate students, and I'll be doing some of these studies on lung TIL, but really the person doing it is Ben Liu in the Hafler lab. I have no timer on this, so I don't know where I am on timing. Someone tell me? Oh, I've got a little time. Okay, good. So let me say one last set of experiments that are being done at Yale, looking at some basic science that could have a big impact on T cell therapies. And by the way, I should point out that, I've mentioned a few people who are doing work, but there are many others doing work at Yale. I don't have time, unfortunately, but I don't mean to leave other people out who are doing really vital stuff that, in fact, probably there are a lot of things I don't know about that I wish I did. So one of the things that Sam Katz's lab has been working on for quite a while, and also Sherman Weissman's lab was working on, is the idea of mRNA reprogramming. So he's using something called CRISPR-I, which is a CRISPR-based technique to knock down genes, but not to actually cause mutations or changes the actual DNA. The advantages of this technique is that you can do multiple RNAs at once. Oops, sorry about that. The other thing about this, of course, is when you do these things by RNA, RNA is temporary. So there are pluses to that and minuses to that. The pluses are that it's a lot safer. You're not permanently altering a cell, okay? The negative, however, is that it's only temporary. So if you want to have effects that last a long time, this might not be the method to do it. But you can imagine situations where using this kind of technique, you could really turbocharge a cell for a short period of time. So for example, we could have a CAR T cell, and you could use his technique to make them particularly proliferative at the time. Add in cytokines, for example, to have them particularly powerful in killing. Stimulators of other things. You could have inhibitors of negative regulators. And in fact, Sam has shown in his lab and Sherman Weissman's lab that, for example, they can, at the same time, using their CRISPR-RNA techniques, to increase IL-2 in a cell and decrease BCL type proteins, which are apoptotic proteins. So again, when you think about what I've talked about so far with, let's say, the City Chen lab, in which they can do multiple different things to design sort of permanent T cells CAR T cells that are particularly powerful, you could also imagine adding in these mRNAs to those same cells and making them turbocharged even more. So there's a huge amount of combinatorial things that we could do to improve T cell therapies. And so there's a lot of excitement for all those things. Last but certainly not least, I just want to acknowledge all the people who've been doing a lot of work. So first, the cell therapy DART. The three docs who do it right now are Iris, Stuart, and I. Alex is our CTTN, is fantastic. Sharon Day is our relatively new, but also fantastic research nurse, and Pavan, our CRA. I'm not, but and then we have just an amazing team here doing regulatory and pharmacy, et cetera. Also, of course, the ACT lab, which is really going to be the people developing. The next therapies that we do here. I mentioned the melanoma team because really we've been doing TIL at Yale for a very long time, and the person who got us started here was Mario Schnoll, and a lot of the ideas that I talked about with regards to how to study these things really came from Mario. Harriet has done the most TIL of anybody here and has done, again, a huge amount. And Sarah Weiss has seen a lot of the patients as well. Katrina Bizak is the person who is really a point person for a lot of cell therapies here. And she's actually also key for setting us up for, as I said, the very, very likely approval of TIL in melanoma. People probably don't know this, but we have our own something called CT Safe, which is for cell therapies. It's a committee to look at really usage and whether we have the capability and the capacity to do all the different trials we want to do. None of this would be possible without the nursing staff on 11 and 12 North, apheresis, Gene Hendrickson, the CRSL, Audrey King, and of course the labs I mentioned. And I should point out that, as I said, we're trying to get an IND right now for lung TIL, and that's based on funding we got from the Lung Sport. And I think I'll leave it at that. Thanks, everybody.
Great presentation. Really excellent. Let's see if there are any questions from the audience. Just gonna check the chat room here. This is from Brooks Leitner. Exciting talk. Have there been any research on adding metabolism modulating molecules or modules in T cells to get around challenging metabolic environment or exhaustion phenotypes?
So there have been there have been a bunch of studies in mice that are really fantastic. Actually, some of the best ones I think were from Sue Keck, who used to have a cancer biology lab, cancer immunology lab here, and now she's at the Salk or Scripps, I don't remember which, but she's in California. But absolutely, so there's no question that in mice you can knock down certain metabolic pathways, making T cells much more tolerant of the toxic microenvironment in the tumor. Now, that hasn't yet been done in people, I don't think, or I should say it's not entirely true. People have been doing screens to make T cells more effective in either CAR T or TIL. And so there might be a company out there that has done that, and we just don't know. But absolutely, that's a huge area of research by a lot of people. And I think we will definitely at some point in the future be seeing CAR T cells that have metabolic pathways altered based on this.
I mean, I think also there's been some recent data looking at diacylglycan kinase and using that as a way of overcoming basically resistance to checkpoint therapy. A number of trials being designed right now. I think it's a very, very interesting metabolic target to look at. And I certainly think this may be one way we could potentially alter the T cell environment. Next comment is from Marcus Boisenberg. Nice presentation, Mike. Just to mention, the Yale Center for Immunology will be striving to support cell therapy efforts, including developing mouse preclinical TIL models.
Yeah, absolutely. I need to talk to you, Marcus. Yes, very excited about that.
And then Jeff Sklar has asked the question, have CAR T studies been performed in patients using multiple CAR T clones simultaneously against multiple different antigens? How many tumor-specific antigens are generally available as potential targets in different types of solid tumors?
So I don't know the answer to that, but I can tell you what I do know. So I think the idea of using multiple CAR Ts at once, I think there's a worry that when you do that, and I think there are data for this, that multiple ones within a cell result in a decrement of the actual response. Like you need to have a lot of the same, you need to have sort of the same CARs activated all at once to really get a good response. If you have too many antigens, it doesn't, I think, work as well. Like you basically dilute out the response. That's what it is. I think you basically dilute it out. Now people are designing right now CAR Ts that are they're heterodimers. So one of the antibody chains is to one target, and one of the antibody chains to another target. That's only going to work if you have really high levels of both antigens, obviously, on the cell. But those are being experimented on right now, and we'll see how those work. There's a real question about if you do that, you're not going to get the binding is just not going to be as good. Regarding the number of tumor-specific antigens, that probably varies from cell to cell. The older studies seem to indicate that, and these are very old studies, so it's very hard to know what that means. But for the TIL studies, in some of the patients, when I looked at the ones who had really good responses, it did look as though there was usually one dominant clone. Sometimes there were two dominant clones. It's hard to know exactly what those data are. It was a very limited number of patients, and it's hard to know if they were looking at the right time. Like maybe the clone was there, did a lot of what it was supposed to do, and then a lot of it disappeared from the blood for some reason. So it's very hard to know how much that's real. The last thing I would say, though, is that it looks as though the most important antigens are private neoantigens, meaning they're not these big targets that we do. And that's really a worry for CAR T cells in general for solid tumors. So that is sort of a separate issue.
Terrific. Mike, as always, great presentation. So I'd like to move on to our second speaker, Dr. Iris Isufi. Dr. Isufi is an assistant professor of medicine and hematology and co-director of the Adult CAR T Cell Therapy program. She received her medical degree from the State University of New York at Stony Brook and completed her fellowship at Yale University School of Medicine. Dr. Isufi's clinical work has been in the area of hematologic malignancies and autologous allogeneic stem cell transplantation for these conditions. As part of the lymphoid malignancy program and transplant teams, she developed a strong clinical interest in aggressive lymphomas. She's focused her efforts in treating patients with aggressive lymphomas as part of clinical trials and consolidating the response to treatment with either autologous or allogeneic transplant based upon the specifics of their disease. As co-director of the CAR T Cell Therapy program at Cellular Cancer Hospital, Dr. Isufi is part of a team that brings innovative new immunotherapy treatments options to patients with certain types of blood cancers. Dr. Isufi.
Thank you very much for having me. Can you see my slides?
No.
No?
Again.
Oh, you know, I have. Sorry, just have to share. Yeah,
Yep, that's great.
all right. Thank you. So my focus today is going to be in cell therapies for hematologic malignancies and what we're doing here at Yale. I'm a clinical investigator in lymphoma and cell therapies. I have a couple of disclosures. So I'd like to update you today on some of the FDA approved indications for cell therapies in heme malignancies, which are growing by the day. Some of our research strategies to improve response rates and prevent resistance to cell therapies. Some of the challenges we're facing clinically and research-wise. And then I'd like to end the presentation by giving you an idea about the work that we're doing here at Yale as part of our immune cell therapy DART for hematologic malignancies, and then some of the intra-institutional research collaborations that we have started to work on. So as Mike mentioned, there's been an evolution in chimeric antigen receptors over time. The ones we're still using in the clinic that are commercially approved are second generation CARs. But there is some innovative CAR design going on, including suicide CARs as a control mechanism for better toxicity management, these dual targeting CARs that express two different antigen-specific CARs, bispecifics, where you have two linked scFVs within one CAR vector, and then these TCR mimic CARs that are important to address HLA-presented antigens, where they're directing the scFv domain against a peptide HLA complex. Initially, the target was CD19 for B-cell malignancies because, as you all know, it's a pan B-cell marker. Its expression is generally restricted to B cells and their precursors, and represented its surface molecules. So it represented a rational target for therapy in heme malignancies. And so all of the agents that are approved for commercial use are directed at CD19. So we started initially back in 2019, the first approval with Tisagen Leclusel in pediatric ALL. And subsequent to that, we've had a series of approvals, including Tisagen Leclusel and Axicabtagene Silolucel for aggressive diffuse large B-cell lymphoma, transformed follicular lymphoma, and then Lysocabtagene Maralucel, where you are giving the cells differently because it's a defined CD4 to CD8 ratio. So there is some novelty compared to the two previously approved products. And then more recently, Brexucabtagene Autoleucel, just in the last year for mantle cell lymphoma. And finally, just a few weeks ago, Axicabtagene Silolucel for relapsed refractory follicular lymphoma. So the response rates that we see with these drugs, particularly in low-grade lymphomas like follicular, are extremely good, with very high overall response rates and complete response rates in pretreated patients. And then in diffuse large B-cell lymphoma and aggressive DLBCL or transformed, the complete response rates have varied anywhere from 30 to 50%, even though the initial overall response rates are very high. So these are still very good outcomes, don't get me wrong. For this group of patients, the predicted long-term survival is typically less than 10%, when they go on to get CAR T cell therapy. So we've really been able to cure a good subset of those patients. But as you can see, we still have a long way to go in aggressive lymphomas because even of the patients who achieve complete remission, only about 2/3 are able to maintain that. But it's very exciting because just in the last couple of years, we now have all of these products that are commercially approved for use and that we're already using here at Yale. So the other rational target was BCMA in multiple myeloma, which is highly expressed on malignant plasma cells. And we know that higher concentrations of soluble BCMA are also associated with poor outcomes in multiple myeloma. This is very essential in regulating B-cell maturation and differentiation. And so there have been a series of phase 1 and 2 studies looking at BCMA-directed CAR T cells, and particularly the first one, idacaptogene biclusel, is actually very close to approval. These were very heavily pretreated patients with a median number of treatments being about 6. And as you can see, the overall response rates are extremely good, and even complete response rates are very good. And so there is, of course, toxicity like we saw with anti-CD19, particularly cytokine release and neurologic toxicity. But again, this is a very difficult population of patients to treat. The majority of them were what we call triple refractory to IMiDs and proteasome inhibitors, and about 25% of patients were penta-refractory, including CD38 monoclonal antibodies. So these are extremely good outcomes for this patient population. And there's now a race to get FDA approval in the USA, not just only for idacaptogene biclusel, but also for several other products. And there are efforts being made to introduce them in earlier phases of disease and comparing them to the standard of care, which is autologous stem cell transplant. And then there are already efforts being made to mitigate antigen escape by combining, for example, BCMA CAR T with CD19 CAR T or targeting other antigens. So the same cannot be said for acute myeloid leukemia, unfortunately, which has been, a great challenge over the years. And because many of the potential target antigens are actually intracellular, they're tumor-associated antigens or neoantigens. And the proteins that are expressed on the surface of the malignant leukemic cells, like CD33, some of those markers are also expressed on hematopoietic stem cells. And so the trials going on have had to consolidate CAR T cell therapy or rescue, I should say, the marrow with an allogeneic stem cell transplant. So there are several critical unresolved issues with CAR T cell therapy in heme malignancies, and I would categorize them in failure to achieve remission, disease relapse, toxicities with CAR T cell, and then some of the challenges in moving beyond B-cell ALL and diffuse large B-cell lymphoma to other diseases that may not necessarily have high expression of surface markers that are easy to target with CAR T cell therapy, or certain diseases where the malignant clone is residing inside a lymph node and not necessarily in the circulation like with B-cell ALL. And so there is that challenge of the tumor microenvironment prohibiting the T cells from getting there. So what is it that predicts outcome from a patient perspective and risk factors that we can outline before they go on to CAR T-cell therapy? So there was this large study that looked at baseline factors that were associated with worse overall survival and progression-free survival in patients who got standard of care axicabtagene ciloleucel. And as you can see here, there were several factors that were statistically significantly associated with worse outcomes. And in particular, I would outline here patients that had a high bulk of disease and patients that had, for example, elevated LDH levels pre-transplant. Patients who required bridging therapy also were at higher risk of having worse overall survival and progression-free survival, perhaps because both of these things are a surrogate for a higher disease burden. And then interestingly, some of the other factors that were associated with outcomes were younger age and also male gender. And that is very different from what we see included in our prognostic indices for lymphomas, where actually older patients tend to do worse. And this means that we need to really relook at our prognostic markers in the era of cell therapy and really redefine what relevant clinical risk factors are. So this is showing a multivariable model of axicabtagene ciloleucel treated patients, where again having a poor performance status and also having high LDH levels is associated with worse progression-free survival and overall survival. And then what about the disease itself? One of the things that we already know is that about 25 to 30% of patients who relapse after CAR T-cell therapy experience loss of CD19. And this was demonstrated in the ZUMA-1 trial and the US CAR T Lymphoma Consortium. So it's not the majority of patients, particularly in lymphoma, but it is a good subset. And so, what can we do to prevent antigen loss and then also this PD-1, PD-L1 mediated CAR T-cell inhibition? And so, because we know that PD-L1 upregulation is actually contributing to CAR T exhaustion. We have this publication, nice publication in Nature Medicine from 2020, where Nirav Shah in Wisconsin actually looked at point-of-care manufactured bispecific anti-CD20 and anti-CD19 CAR T cells in relapsed malignancies. And some of these patients had already undergone anti-CD19 CAR T cell therapy. And they do see ongoing responses in about 40% of patients. I think out of about 60% that responded initially, and they did not observe loss of CD19 in progressing patients when they targeted the tumor with the bispecifics. So really very, very exciting data. And then just this year, just this past year at ASCO, they presented results of a first CD19 and CD22 targeting bicistronic, which is dual antigen targeting with humanized binders to reduce immunogenicity. And in addition to 4-1BB costimulatory, they also added OX40 to improve persistence. So based on that early data, they went on to do a single-arm, open-label, multi-center phase 1/2 study where they did dual targeting of CD19 and CD22, but they also added pembrolizumab for relapsed refractory diffuse large B-cell lymphoma. And interestingly, what they saw is a high rate of complete responses, about 66%, although it's too early to say, how, if they are going to be durable and how durable they will be, because right now they only have short-term data. But interestingly, there was very little toxicity with this particular construct. They did not see any grade 3 or 4 cytokine release or neurologic toxicity. And that perhaps really is a reflection of this really novel technology that they're using with a novel pentameric spacer and these humanized binders. So this data is very exciting because it's a therapy that we might be able to use, if approved, eventually in the outpatient setting and delivered in the outpatient setting, and that's where they're really going with this. So what else do we know about the disease aspect itself that may make a response to CAR T-cell therapy challenging? So this data from the Juliet study, which was the global phase 2 trial of tisagenlecleucel in relapsed refractory diffuse large B-cell lymphoma, they looked at the MYC expression and tumor infiltrating T cells in that study. And what they actually found was that a baseline MYC negative status was actually associated with a significantly improved outcome compared to MYC positive patients. And that included also longer median duration of response and overall survival. And when they looked at the tumor microenvironment analysis of the baseline biopsies, what they saw is that lack or low frequency of tumor infiltrating CD3 positive T cells was also associated with short progression-free survival compared to patients that had more than 3% CD3 T cells in the tumor. So taken together, these results suggest that MYC overexpression or an unfavorable immunosuppressive tumor microenvironment with a restricted T cell response may impact CAR efficacy in patients with large B-cell lymphoma. And then this publication in OncoTarget looked at mutations or copy number losses of CD58 and TP53 genes in diffuse large B-cell lymphoma and showed that these are independent, unfavorable prognostic markers. So what we know about CD58 is that this is actually binds CD2 on the T cells, and T cell mediated cytotoxicity and also NK cell mediated cytotoxicity is actually quite important and quite dependent on the expression of CD58 on the tumor tissue. So in ASH 2020, they presented data looking at CD58 mutations in circulating tumor DNA, and they showed that this was associated with poor outcome after axicabtagene ciloleucel. So these CD58 mutations are or loss are common, and they occur in about 20% of patients with diffuse large B-cell lymphoma. And then in addition to that, the protein CD58 protein expression is also deregulated somewhere between 60 to 80% to 70% of patients with diffuse large B-cell lymphoma have deregulation of the CD58 protein expression. And as you can see here, they were able to show that loss of this expression was also associated with worse outcomes, where in blue here you see CD58 wild type and in red CD58 alteration. So fewer patients that had loss of CD58 expression actually went on to achieve complete remission. The majority either did not respond or they achieved only partial remission, and then they went on to progress, unfortunately. So Meisner group presented this data very interesting this year at ASH, where they showed that integrating CD22 co-stimulation within CARs was actually able to overcome CD58 loss in tumor cells. And they tried this both in cis and in trans, and it wasn't until they integrated it in trans that they saw these results. So this was very eye-opening for us because we used to think that all of the co-stimulation is coming from other cells, and we didn't really realize how important actually CD2 was in CAR-mediated cytotoxicity. So CD58, CD2 was a very novel axis of CAR resistance that was uncovered through deep correlative studies in patients getting cell therapies. And CD58 loss or mutation portends a poor outcome, but perhaps we can overcome that by engineering these CARs that integrate CD2 signaling in trans. And this is important because CD58 loss and mutations are also common in other cancers and are likely able to mediate resistance to other CARs and immunotherapeutics. So it could perhaps be applied in other malignancies outside of diffuse large B-cell lymphoma. So I've spoken to you about the relapse refractory setting, but we are now doing studies pushing these cellular therapies in the second line and even in the first line setting. Zuma 12 looked at very high risk patients with high-grade B-cell lymphoma with MYC, BCL2, and BCL6 translocations. And they did PET-directed therapy. And for patients who still had disease after 2 cycles by PET scan, they actually went on to get their T cells collected and then receive CAR T cell therapy. So these are the results. They saw a very high 85% overall response rate with 74% CRs. This is a difficult group of patients for us to treat because oftentimes they do not achieve remission and they progress right through therapy. And then interestingly also, or maybe as expected, the CAR T cell expansion was greater in this study when compared to Zuma 1, where patients with relapse refractory disease were treated. So higher quality T cells with higher proliferation and higher expansion. So this has not, obviously it's not prime time for us to change our decision making and move this to first line therapy, but there is definitely improved T cell fitness in first line treatment. And this may be the wave of the future when we get more long-term data. So just to sort of recap for you some of the studies in relapse refractory disease and also to include some data with CLL and mantle cell lymphoma. As you can see, very high overall response rates across the board, and then somewhere between 50 and 75% complete remission rates in relapsed patients. So where are we going with this? As I mentioned, we're trying to introduce them earlier in the lines of therapy. So many phase 3 studies looking at second line for transplant eligible patients, randomizing them to transplant versus CAR T, and then perhaps eventually in the front line. And then in ALL patients, looking at patients who are MRD positive after 1 line of therapy. And then hopefully some of this phase 3 data in adults will result in an approval, because we still don't have an approval in ALL for patients over the age of 25. So what about allogeneic CARs? As you know, there are some limitations with autologous CAR T cells, particularly in terms of cost, harvesting and manufacturing failures, and disease really progressing during manufacture. And we can really bypass a lot of that with donor-derived cells, where we can really reduce the time to infusion significantly and actually be able to take more patients to CAR T. And there's an increased probability of healthy CAR T cell generation and the convenience of repeat dosing if necessary. So these are some of the investigational allogeneic CAR T cells for heme malignancies, targeting different antigens, both in lymphomas, ALL, but also in multiple myeloma. This is still early phase 1, phase 2 data, but I think that this is going to be the wave of the future in CAR T. So I'm going to now shift gears to just briefly talk about our CAR T-cell therapy program here at Yale. We started our efforts in 2018 and were able to eventually treat our first patients in January of 2019, and then we're able to actually achieve FACT accreditation after extensive auditing of our program. So this is our organizational chart. As you can see, it includes collaboration between multiple different departments, physicians, nursing programs, cell therapy with Diane and Alexi, and also apheresis with Ed Snyder and Jeannie Hendrickson. We have a really trained group of people being able to apherese the patients and then give the conditioning therapy and manage the toxicities. So what have we done in the last 2 years? We've treated 57 patients, some of them with axicabtagene ciloleucel and some with tisagenlecleucel. We're just starting to actually expand to mantle cell lymphoma and then follicular lymphoma. And we've also treated 11 patients on clinical trial for multiple myeloma with anti-BCMA mRNA CAR T cells. And as Mike mentioned, there's much less toxicity, but there are also challenges in terms of the short life of the mRNA. And perhaps the need for frequent dosing or maybe introducing this in earlier lines of therapy where patients are not very heavily pretreated and do not have an extensive burden of disease. With the new approvals in multiple myeloma expected this year, there's actually an anticipated significant rise in numbers of patients that were going to be treated. And what that means is that we can collect a lot more data and do a lot of studies on patient samples. So this is the Yale Advanced Cell Therapy Lab. And then this is our immune effector cell therapy DART that Mike and I co-lead, and we have a team as he spoke about, so I won't belabor this, but we wouldn't be able to do what we do without their amazing work. So this is our portfolio. We have some studies that were opened and finished accrual, but as you can see, we have a large number of pending studies, the majority of which are very novel because they're either bispecific CARs or they're allogeneic CARs, CD19 NK CARs, really also these randomized comparative studies introducing CAR T cells in the earlier lines of therapy. We really took a setback with COVID, but we really hope to be able to open all of these studies in the next few months and start enrolling patients. So this is these are some of our intra-institutional research collaborations with Dr. Mina Xu and Dr. Jordan Pober in pathology, looking at the vasculature in the human lymphoma nodal microenvironment. Collaboration with Xiaolei Xu, looking at these phase CARs for low antigen expressing B-cell cancers, and then collaborations with Sidi Chen. So in the interest of time, I will just briefly discuss these collaborations, but as you know, getting the T cells to the tumor tissue and increasing homing is actually quite a challenge for most patients. And there have been several attempts over time looking at how we can improve homing for lymphocytes, including cell surface painting, for example, to insert alpha V beta integrin into primary lymphocytes, including glycoengineering CAR T cells, for example, to enforce E-selectin binding, because as many of you may know, CAR T cells do not express sialyl Lewis X and do not bind E-selectin, but we can actually enforce their display on human CAR T cells by surface fucosylation, and this results in very robust E-selectin binding, even under conditions of hemodynamic shear. And then also gene therapy using genetically modified lymphocytes targeting VEGFR2 in highly vascularized tumors. But unfortunately, all of this data is in mice, and we don't really know what's happening in the human tumor vessels, and we do not have any idea about the spatial relations of these tumor-infiltrating lymphocytes. And so the aim of our study is to employ highly multiplexed immunofluorescence imaging of human lymphomas to spatially correlate and phenotype the infiltrating immunocytes using formalin-fixed and paraffin-embedded tissue specimens. And then we want to apply these results to investigate the lymphoma vasculature in CAR T patients pre-treatment and post-treatment. This is Nathan Paulson, one of the residents in pathology, and he's already looked at some of our lymphoma tissue samples, showing that there are differences in expression levels of vascular adhesion molecules, for example, between diffuse large B-cell lymphoma, classical Hodgkin lymphoma, and T-cell rich large B-cell lymphoma. And so we want to do high dimensional phenotyping of these vascular cell in FFPE, de-identified tumor samples, looking at all of these vascular markers. And we anticipate to find some correlation of the vessel phenotypes with the abundance and phenotype of the leukocytic infiltrates, and to correlate this with the patient's outcomes post-CAR T-cell therapy. And if we're lucky, we're going to be able to show that, a rationale for combining these CAR T-cell therapies with anti-angiogenic therapies, particularly, and this can be a launching point for us to actually eventually in the future consider a trial. So this is some of the data from Xiaolei Su's lab, where he's using these phase CARs where targeting specifically low density surface antigen, and he's been able to show that CAR signaling is different from T-cell receptor signaling, and that it bypasses certain proteins like LAT and has a different pathway that results in actin polymerization compared to TCR signaling. And he's actually using this information to develop these phase CARs on lipid bilayers that he can modulate to recognize low density surface antigens. So this is, again, some of the data that he has generated in his lab, where he's been able to show that CAR T signaling bypasses this important scaffold protein promoting phase separation. He's been able to build these phase CARs where they contain a new control modality that can leverage domains affecting phase separation to modulate CAR T activity, recognizing low density surface antigens. He's constructed Raji B cells expressing low to high CD19. And this is just some very preliminary data that he has showing that these phase CARs display superior activity compared to control CAR Ts against low CD19. So we are hoping to look now at some of our patient blood samples that have low CD19 expressing malignancies either at baseline or following treatment with CD19 CAR T cell therapy, and showing how these phase CARs will be able to act against these low CD19 expressing tumors. And then we're also hoping in the future to collaborate with Chen, looking at these dual knock-in, knockout CAR T cells that he's engineered in his lab, targeting 2 different antigens on lymphoma cells and doing PD-1 knockout. So this is our group, and really dedicated group of people, and I'm very thankful for their work. And I went a little over time, so I'm happy to answer any questions.
So I don't think there are any questions on the chat room at this point. So Iris, thank you for a terrific presentation. I guess we'll close
Thank
out.
you.